Yeah, thanks for the introduction, and thank you to the steering committee for inviting to give an overview of the state of water quality science and how it links with Cots outbreaks. So this talk is really a review of the work that's been going on in that space and what we're proposing to do, rather than just only my own work. So this beautiful picture that you see here, many of you would have seen it already, is the Frederieke River in flood in 2019 when Townsville went under, and you can see that that particular flood plume is huge. In this case it did reach mid-shelf reefs, and later on it even reached some of the outer reefs, and later on I'll show you some animations about how far those flood plumes travel up north as well. So as some presenters before me already stated, the drivers of outbreaks, they're still being actively debated. There's probably consensus about that there's multiple drivers acting at any one time, or more than one, different combinations, different locations. Different things may be going on, but water quality has definitely been one right from not quite from the start, but from the early beginnings of Cots outbreaks on the Great Barrier Reef as potentially being one of the drivers of these outbreaks occurring. It's also one of the three management approaches on the Great Barrier Reef, so there's a lot of investment and effort going into water quality improvement, alongside with manual control that you've heard a lot about, as well as zoning. So why water quality? There's the hypothesis around it, and Sven already referred to it, is the nutrient enrichment or terrestrial runoff hypothesis, which states that enhancement of nutrients in coastal waters, or I shouldn't say only coastal waters, in reef waters, drives phytoplankton blooms, which is a food resource for Cots larvae. If they have more food, the idea is that they are in better condition, more survive, more can settle, and then voila, you have an outbreak. Of course, it's not that simple, but the idea is that extra nutrients do, through these various steps, contribute to Cots outbreaks and may even be one of the main drivers. So in this talk, I'll give you a brief overview of each of the steps that we've identified should hold if water quality does in fact drive Cots outbreaks, and then close off with a summary slide about the effectiveness of water quality improvement so far and influencing Cots outbreaks. So as I said, for the nutrient hypothesis to hold, we think there should be 6 steps that link changes in land management, or at least for the terrestrial nutrient runoff hypothesis to hold. There's 6 steps that link changes in land use and management to eventually Cots outbreaks in step 5 and 6. Now, I should acknowledge my colleagues at AIMS. This was led by Sven, this diagram, together with Barbara Robson, myself, and a few other colleagues, to basically get a structured understanding of how this may work. So we developed this a couple of years and then structured our thinking around what are still the key gaps in research. And a lot of those have also been identified and articulated in the SIP program, as you would have heard yesterday. So I'll briefly go through each of these steps, which basically link land use change and management into increased river pollutant runoff into the reefs. That results in increased nutrient concentrations in reef waters, resulting in increased phytoplankton. Increased phytoplankton is a food resource for Cots larvae. You get more Cots larvae that settle, and you get a Cots outbreak. So each of these steps I'll briefly go through and see what evidence we have for that and where we still have gaps. So this is the first one. This is actually a pretty easy one, where I show here 2 plots of land use in the catchment that discharges into the Great Barrier Reef, with land use before European settlement and land use as it was probably around 2014. These maps are a bit out of date, but land use wouldn't have changed too much on a large scale during the last 10 years or so. Main point, what you can see is that there was a lot of forest before European settlement, and now the main land use is cattle grazing, the pinkish colours on the right-hand side. What that has resulted in or contributed to is that we now have an increased runoff, or sorry, I should say pollutant loads into the rivers that discharge into the Great Barrier Reef of sediment, nitrogen and phosphorus compared to what it used to be. And that differs from region to region, differs from river to river, but overall on the Great Barrier Reef, those are the increases that we have estimated to have occurred based on modelling exercises and monitoring exercises. Now, the reef is not unique in that respect. We see this all around the world where catchment land use and management has changed. River pollutant loads generally increase unless you put a big dam onto it, and water quality in coastal waters deteriorates. So really no more questions in this space, although probably if I talk to catchment managers, there'll be a lot of questions, but in the Cots space, let's just say, yes, enough evidence there. So the second step then is, well, we have this increase in runoff into coastal waters. Does that also then link to increased nutrients presence and availability in coastal waters? And what I show you here is an animation that Barbara Robson did for me, especially for this presentation, that shows flood plumes from the wet tropics rivers that flow into the initiation zone between Cairns and Cooktown. And so what you see there, the different colours are different salinities, so basically showing freshwater input from those rivers into the initiation zone, where we think nutrient input is particularly important for initiating outbreaks. So that's freshwater, but before I go then to nutrients, this is the Burdekin, and that's a really interesting one, and related back to my title page, whopping big flood plume coming out, and freshwater plumes travelling right up into the initiation zone and possibly even further. Now that is not salinity, that shows proportion of Burdekin floodwaters into those coastal waters. So when it's really dark, that's 10% of the water is Burdekin floodwater. So when that river goes, it goes a long way, and it can definitely contribute into that initiation zone. Question then, of course, is, well, does it matter, and does it bring nutrients into that initiation zone as well, or has it transformed nutrients as it travels up and turn into phytoplankton blooms? Let me just see. So yes, direct monitoring of flood plumes as part of the marine monitoring program that Mike referred to earlier definitely shows that we have increased levels of both sediment and nutrients, but particularly nutrients in flood plumes. And but in mid-shelf reefs and offshore waters, it's difficult to determine because these nutrients get taken up and remineralized very quickly. So by the time waters get to those mid-shelf offshore waters, because it's warm, a lot of sunshine, it gets taken up very quickly, especially nutrients that are highly bioavailable. But in flood plumes itself, yes, definitely we see higher levels or higher concentrations of nutrients. And then have those nutrients increased since land use changes in land use and management? We can refer back to coral cores, where we can go back in time and look at signals of fresh water, sediment, nutrients, nitrogen and phosphorus, and we do see clear signals for some of these when land use changed about 100, 200 years ago. That is still debated to a certain extent as well as to how exact those signals in coral cores are, but we definitely see some changes over time there. So yes, there is a runoff signal. Now one of the key questions here still is nutrient availability, presence and availability, does that increase in the initiation zone itself? And if so, which nutrients are they? And that's an important question because the wet tropics rivers generally deliver more inorganic nutrients related to fertilizer use, whereas the rivers further down south, like the Herbert and the Burdekin, they generally deliver a lot more particulate nitrogen and particulate phosphorus makes up a much larger proportion of the nutrients in the flood plumes. Now, generally the thinking was, well, they're not bioavailable anyway, so who cares? But actually some recent work shows that those particulate organic materials do get transformed into reef waters quite quickly and actually play an important role in driving or in supporting plankton blooms. So we should really be considering rivers further down south as well. And to some extent, that's already been done in the investments for water quality improvement. So then the next link is, well, we have increased nutrients in reef waters. Does that then result in increased phytoplankton presence, abundance? Do we get blooms? You only need to look at some of these satellite images and you would say yes, but of course we need to measure things as well. And surprisingly, little work has been done on phytoplankton in the Great Barrier Reef. So there's some earlier work that has looked at presence and distribution, as well as some work on dynamics, including the role of nutrients by Miles van Nassedel in 2005. But since then, very little work has been done to describe the response of phytoplankton species and community assemblages in the Great Barrier Reef. And Sven referred to, his samples of larvae and plankton samples, which would be great to inform which species may drive the presence and abundance of Cots larvae. And so what I'm showing here is actually some very recent work, again, back to the Burdekin 2019 flood, where Barbara Robson led a project to look at phytoplankton presence, abundance, and species composition right immediately after the flood, or as immediate as it could be, because we couldn't go anywhere when the flood was happening, as well as in winter in June, July. And you can see every dot there is a sample. The summer community assemblage is very different from the one in the winter. It's much more diverse as well. And when you look at the arrows, they are driven definitely by some of the nutrients present in the water, like nitrate and silicate, as well as salinity. So, and all of these vary between flood plume conditions and not flood plume conditions. So certainly we do see a response in the phytoplankton community assemblages. Whether those include plankton species that Cots larvae like and thrive on, we don't really know yet, and that's an important gap to investigate as well. And importantly, do all these phytoplankton species, or not necessarily these, but ones that Cots larvae may prefer over others, do they occur in the initiation zone? Holy crap, 5 more minutes. All right. Increased phytoplankton, does that lead to increased recruitment of Cots larvae? I probably already referred to that a bit. We don't really know. We certainly know that they overlap in a spatial context, and you see two plots here from two different studies. Flood plume satellite image from 2007, which shows green waters right up and down the coast, including the initiation zone. All that green water is full of chlorophyll A, which translates into phytoplankton, although we don't know which species or assemblages. On the plot on the right, Sven already presented that, but that shows Cots larvae also shows variation in spatial occurrence. Do they overlap in time? We don't really know. Of course, it's important that if this hypothesis holds, that the food resource needs to be available at the same time, same space as Cots larvae, so during the spawning season in December and November. All right. And then 5 and 6, I can be fairly quick about this one because there's really no reported evidence for Cots on this one, and I was very pleased to see Peter's presentation before lunch, because that basically touches on at least one of these steps, where we're looking at whether higher larval recruitment results in higher settlement rates and then consequently result in higher adults and outbreak dynamics in the initiation zone and or further downstream. And so it seems self-evident, but not necessarily so. And so work around looking at settlement rates and influence on adult densities is certainly something that is a gap and has been identified in SIP as well. Also, can other drivers be involved in driving these relationships, such as predator presence or even food resources for Cots? Coral cover, for example. If there's no coral cover, then they're probably not going to outbreak either. So a whole range of gaps still all along the way, and many of these are at least being put forward by SIP to be addressed. Then to wrap up, efficacy of water quality improvement so far specifically in a Cots context. So a lot of policy and investment has been taken place to improve water quality in the reef, not only for Cots, but Cots is certainly a major driver for that. And this pretty much started in 2003 with the Reef Water Quality Protection Plan and has evolved through various plans into the Reef 2050 plan. And a lot of investment has been gone into by the Australian and Queensland government, up to about $1 billion since 2009. So has it resulted in improved water quality, which we would like to have if the Cots outbreaks hold? Almost done. So when we look every year, water quality improvement gets reported on in annual reef report cards, and pretty much inshore water quality gets a score of moderate to poor, or poor to moderate. It varies from year to year. The most recent one was poor again. So that doesn't mean nothing is happening in the catchments. There's certainly progress there, and we can see some improvements in some river pollutant loads, although in the scheme of things, they're probably still fairly small. Things are happening though, and there's certainly legacy effects of finally detecting some trends in water quality improvement in the reef. Modeling studies definitely show that it's possible, and we have some good international examples that it's possible as well. So a recent study on Chesapeake Bay, which is about half the size of the reef catchment, found that after 30 years of effort, they finally could detect measurable improvements in water quality, 23% drop in nitrogen, nitrogen concentration. So it definitely is possible, but it's a long-term haul. So for Cots then, that means that water quality improvement at the moment is not really influencing Cots outbreaks yet. We actually have an outbreak as we speak. And how long will it take before water quality improvement will actually influence Cots outbreaks? That's an important question to ask, particularly in the management context, because at the moment we've got manual control, water quality and zoning, and what relative role can water quality play in that space now, or a couple of years, or a couple of decades. And I'll leave it at that because I'm already over time. So thank you very much.